Welcome to this video where we want to be reviewing what's happening with vaccine development as we come towards not too far off the end of May now. So where are we with vaccines? Because vaccines, when we get a vaccine for the COVID-19, that is essentially our way out of this crisis. Things can go back to normal. We can do a lot between now and when we have a full vaccine, but the vaccine is going to be the ultimate escape, I believe, from this problem. So where are we with vaccination? And I've got a couple of groups I want to report on this morning that seem to be leading in this matter of producing vaccine against the SARS coronavirus type 2. Now, this first one I want to tell you about is the Oxford Astrazeneca collaboration. Now, they've started actually human trials already, but they've actually just reported on some trials from rhesus monkeys. Now, I probably don't like animal research any more than you do, but this is what's happening, so we just have to report it. Now, what happened was they vaccinated 6 rhesus monkeys with this new candidate vaccine they have developed. And they gave all the monkeys 2 doses of the vaccine. And the hope is that the monkeys will develop antibodies and therefore be immune and therefore be vaccinated. But let's look at what actually happened. So all of the 6 monkeys in the Oxford University Astrazeneca trial, they were all vaccinated, treated with the Oxford vaccine, but then when they became challenged with the actual virus, they all got infected. So what this means is these monkeys were given this potential vaccine, 2 doses, allowed time to develop immunity, and then they actually had the virus, the COVID-19 SARS coronavirus 2, put in their noses, and they all became infected. So on the face of it, that doesn't sound too good. It sounds like the vaccine is not working at all. And the viral loads in the noses of vaccinated and unvaccinated animals were identical. So monkeys that were vaccinated developed the virus, and monkeys that weren't vaccinated developed the virus in just the same way. Identical viral loads in their noses. But when it came to the lungs, there did seem to be a difference, because the monkeys that were vaccinated had less severe clinical features than the unvaccinated monkeys. And of course, this is what matters, that we are not as sick, preferably not sick at all, but not as sick. So the vaccinated monkeys were not as sick, even although they were both expressing the virus in the same numbers. So it looks a bit like this vaccine might have been protecting the lower airways more than the upper airways. It's hard to see how that could be, but this is what the reports show. Now, what we want is neutralizing antibodies. We want neutralizing antibodies. Now, as you know, the antigen is the virus, or in this case the vaccine, what the body recognises as foreign, and in response to that the immune system makes the antibodies. And it's the antibodies that actually give us the immunity. These are the immune proteins. And then these spikes on the end of the virus that allows the virus to get into the cell, the neutralizing antibodies will put a cap on that and make that useless, so the virus can no longer invade cells. These are the neutralizing antibodies that we want. And for some strange reason, we measure antibody levels in what we call titres. So that means the levels that were generated in the blood and the tissues, really. So this vaccine didn't develop that many neutralizing antibodies. And yet the vaccinated monkeys were less sick and had less pneumonia. So they also noted that the viral loads in the monkeys' noses were still enough. There was still enough virus in the monkeys' noses to cause infection. In other words, the neutralizing antibodies didn't get rid of enough of the virus to stop the monkeys being infected. So insufficient antibody titers to prevent viral shedding in nasal secretions. In other words, these monkeys could still infect other people or other monkeys. They were still infectious. So on the face of it, not looking brilliant, not quite what we'd hoped for. And at the end of the day, the vaccine that we get might not be a perfect vaccine, but it'll be something. It'll be a lot better than nothing. So if you could vaccinate everyone and stop them getting severe disease, that would be a lot better than not vaccinating people at all. So anyway, let's go on. Now, the human trials actually began with this vaccine in April. And they had a vaccinated group and an unvaccinated group. So just the same as they did with the monkeys, where they had some that received the vaccine and some that didn't receive the vaccine. They've done the same with people. Some have had the vaccine and some have not had the vaccine. So they've got a control group. And at the moment, the group are waiting for the vaccinated people to develop the appropriate longer-term antibodies. Now, the fact that the group are carrying on with these human trials, to me, means that they're probably optimistic. So what the group are expecting, and absolutely what the group are hoping for, is that the group that were vaccinated, the group of human beings that were vaccinated, will develop lots of antibodies, whereas obviously the group that weren't vaccinated, you wouldn't expect to develop antibodies unless they've been exposed to the disease. And the results from this will be there in a few months. It takes time. It takes time to develop immunity of the longer term types of immunity, and it takes time to do all these things. So it's not happening that quickly, unfortunately. So within a few months, we'll know if this Oxford University Astrazeneca vaccine is producing neutralizing antibodies in human beings. And another thing that makes me think they must be optimistic is they're now preparing for the next phase of the phase 2 clinical trials, involving 30,000 patients. Some of these are going to be children, some of these are going to be older people. So they're now preparing for a much larger human trial. So I feel they must have grounds for optimism to be carrying on with this. And the thing is, they may well believe at the moment that this vaccine's safe as well, otherwise I don't think they'd be carrying on with 30,000 subjects, because there's a risk with vaccines. New vaccines are like any other drug. They can do harm as well as doing good. So new vaccines risk antibody-induced enhancement. Now, what this means is the virus makes you sick when it actually gets into the cells. Viruses are obligate intracellular parasites. They only work inside the cells. And to get into the cells, they have to get into the cells somehow. So the virus has this way of getting into the cell. But some antibodies will actually make it easier for the virus to get into the cell. So some vaccines can actually make the infection worse. And this is what this antibody-induced enhancement means. So we don't want that. But why are we worried about this? Well, this effect, this antibody-induced enhancement, making it easier for the virus to get into the cells, caused serious lung damage in animals given experimental vaccines for both SARS-1 and Middle East respiratory syndrome, which of course are both the two previous serious coronavirus infections that the world has dealt with. The SARS-1 was 2002, 2003. The Middle East respiratory syndrome, I think that started in about 2011 and still crops up occasionally as the virus can spread from camels. So there was a vaccine developed for these that actually made the condition worse. So we have to make sure that's not the case. But the good news is they're cracking on and they are preparing for phase 2 clinical trials with human subjects. And of course, monkeys and humans aren't the same. So it would be a bit surprising, but it could be that you had an absolutely brilliant response in human beings and an absolutely rubbish response in monkeys. Now that would be unlikely because we share quite a lot of DNA with rhesus monkeys. But who knows? We don't know. We don't know till the empirical work is done, till the experiments are done. So they are carrying on with that at full speed and they have been well funded. So money's not a problem there at the moment. Now, this is the second project. This is the Beth Israel Deaconess Medical Center, which I believe is in Boston in the United States. Now, references to look at as always. Now, they report 2 studies. So I'm going to talk about both of these studies because they're both very interesting. And the results of both of these studies are remarkably encouraging, it has to be said. Infection prevents reinfection. Right, so they've established that. I'll tell you how I'll tell you why they've given more evidence for that. Now, we've had this question mark for some time. If someone gets the virus, can they get it again? Can they get it twice? Will having the illness and having the virus, will the body recognise that virus as an antigen and generate sufficient antibodies to stop you getting it again, to make you immune? And the answer seems to be yes. That seems to be the answer. And I've got two reasons for saying that. I'm going to give you this one reason in a minute. But the other reason was the data we looked at from South Korea just a couple of days ago, where people were testing positive. A few people, not many, a few people were testing positive for the virus after they'd been ill. But it was found that these were false positives. They were just viral residues. And the virus was dead. It wasn't capable of reproducing. So empirical human data from Korea is indicating very strongly that once you've had this infection, you should be immune, you shouldn't get it again. And that was a study of about 290 people in South Korea. And of course, that was from over 10,000 people in Korea that have been infected. So that's looking very promising. And this also supports that, as we'll see. So they found that out. Then prototype vaccine protects against SARS-CoV-2. So that was the next bit. They reckon that antibodies do provide protection. Now, I put these together because if you have the virus, you will develop antibodies. And if a vaccine is effective, you will also develop antibodies. The difference is that if you're vaccinated, you won't get sick. But this study shows that again, it's in monkeys, this study. So the monkeys that had the virus were protected, and the monkeys that had the vaccine they developed were protected as well. But let's look at that in a bit more detail. So study one. Now, study one was nothing to do with vaccines. Study one was just seeing if monkeys who've had the infection develop neutralizing antibodies and are protected from reinfection. That's the research question. So nothing to do with vaccines at all, study one. So what they did was they injected 9 rhesus monkeys. Well, not injected, they inoculated them. They put virus into their noses and their pharynx that would go into their lungs. So in other words, they gave the monkeys the disease, which, okay, is not a nice thing to do, but that's what they did. So the monkeys were inoculated. And when I say inoculated, I mean they were given the live antigen. So to me, inoculation means giving the live antigen as opposed to a vaccination, which is giving an attenuated, a weakened antigen that won't cause disease. So they took the live virus and they infected 9 monkeys with it. Now, the virus quickly spread in the monkeys to the upper and lower respiratory tracts, as you would expect. So the monkeys basically developed COVID-19 disease, as you would expect. And unfortunately, all of the 9 monkeys developed viral pneumonia. And again, this isn't entirely surprising because I think the monkeys were given quite a large inoculating dose, and all 9 monkeys developed viral pneumonia. In other words, all 9 monkeys that were given the virus developed SARS coronavirus 2, COVID-19 pneumonia. But all the monkeys recovered after 28 days. So they all got better after 28 days. So that's good. Now, a week later, after they got better, they re-exposed the monkeys. So once they got better. And none of the animals got sick. So in other words, all of the animals that were sick the first time, when they were exposed to the virus a second time, none of them got sick. So they were all protected. They were all protected. None of the animals got sick. And all of the animals also, when they looked at the blood, all of the animals had protecting neutralizing antibodies. So the illness had stopped them getting the illness a second time, and it had also produced the antibodies, which it would make sense to infer are generating this immunity the monkeys were now enjoying. And this does give rise to the possibility of immunity passports. So we could say some people have had the disease and won't get it again. We're not quite at that stage yet, but we are getting towards that stage now. Now, the second study was in 35 monkeys. And what they did here was that they vaccinated 25 monkeys and they gave them 2 doses of vaccine. But they kept a control group of 10 monkeys that were not vaccinated. So there was a group of 25 monkeys that had the vaccine. Then there was a group of 10 that didn't. 2 groups. So vaccinated, non-vaccinated groups of monkeys. That's what they did. And there've been this group, this Beth Israel group, they've been developing slightly different approaches to vaccines. So they've developed 6 prototype DNA vaccines. Now this is interesting because normally you would expect an RNA vaccine to work because this SARS coronavirus 2 is an RNA virus. The genetic material is ribonucleic acid, not deoxyribonucleic acid as it is in you. But nevertheless, they were the types of vaccines they were developing. And they tried 6 different prototype viruses on these 25 monkeys that they put into the vaccination group. So presumably they got several monkeys with one type of vaccine. They would not have given any one monkey more than one type of vaccine, I'm sure. So all prototype vaccines use the genetic code for production of the proteins that SARS coronavirus 2 uses to invade cells. So that makes perfect sense. So we have this wretched virus, and it's got these spikes sticking out of it like this. And there's a protein on the end of these spikes like that. And it's this protein on the end of the spikes that fits into a receptor on a cell surface and allows, if that whole thing is, it allows the virus to get into a cell. That's how this works. So let's look at it on a different scale. If that's the cell, if that's the human cell there, then the virus comes along here, sticks onto this receptor site, sticks its genetic material in, and the virus reproduces inside the cell, produces lots more viral particles which then come out and are free to go off and infect other cells. So all the vaccines they developed were developed to neutralize these surfaces so it couldn't bind and get into the cell. In other words, it's altering the shape of the key so it can't fit in the lock. I think that's the simplest way to put it. So all of the vaccinated monkeys developed antibodies. No, that's not under it. All prototype vaccines use genetic code for the production of proteins. So that's what. So they're all attacking these what we call adsorption sites for the way the virus gets into the cell, messing up these surface proteins to stop it getting into the cell. Now, all of the vaccinated monkeys developed antibodies, some at recovered levels. So all of the vaccinated monkeys developed antibodies. And some of the monkeys developed so many antibodies, they had the same level of antibodies that the monkeys had had in the study 1, who'd actually had the disease. So there are 6 candidate vaccines, all developed neutralizing antibodies. So this is remarkably encouraging. Now what they did then was 3 weeks after the second dose of the vaccine. So these monkeys, the 25 monkeys, had the vaccine. Then 3 weeks after the second dose of the vaccine, all monkeys were inoculated with COVID-19. So they showed that these vaccines were working in theory by looking at the antibodies. But then they did the experiment, and all of the monkeys in the group, all 35 actually, all were inoculated with the virus. So the 25 vaccinated monkeys were given the virus to see if they got the disease, and the 10 monkeys that were not vaccinated were given the virus to see if they got the disease. And none of the 25 vaccinated monkeys developed high levels of virus in their lungs. So again, critically, we see the lungs were protected. So very encouraging. All of those. So even with just 6, let's see if this particular prototype vaccine works. They all worked to the degree that none of the vaccinated monkeys developed high levels of virus in their lungs. That's pretty encouraging. All the 10 of the unvaccinated monkeys did. So all 10 of the unvaccinated monkeys got viral pneumonia. Or 25 of the unvaccinated ones didn't. And it looks like some of the candidate vaccines, as you would expect, were working better than others, because 8 of the 25 vaccines demonstrated no detectable virus. So 8 of the monkeys that were vaccinated demonstrated no detectable virus. The virus didn't take root in them at all. And presumably the researchers kept a note of which particular prototype vaccine these 8 were given, so they'll know which ones were most effective. So 8 of them developed no detectable virus, having been exposed to the illness after they've been vaccinated. 17 showed low levels of the virus. So what they'll do is they'll take these vaccines that were given to the 8 that developed the strongest immunity and concentrate on those. So that is very encouraging. Now, there's another 2 things here. Higher antibody levels were linked to lower viral loads. So the higher the amount of antibodies, the higher the amount of protective antibodies the monkeys had, the lower the viral load detected in those monkeys after they were reinfected. So they demonstrated it was the antibodies that were highly correlated, negatively correlated actually, with the viral load. So the higher the antibodies, the lower the viral load. So the antibodies did seem to be doing the trick that they're supposed to be doing. So that's encouraging. And what that means is, again, this immunity passport idea, lab tests could predict how immune someone is, because the antibodies were doing their job. So if someone had high levels of antibodies, it could be assumed that they have high levels of immunity and therefore would have low levels of virus and therefore not get sick. So that is all remarkably encouraging. But of course, this is only at animal trials stages. It needs to go on to human trials next, but it's encouraging. So does this all apply to humans? I haven't got a clue. Nor has anyone. We don't know. This is monkeys. Does this all apply to humans? In my view, probably. Probably. I will put it a lot stronger than possibly. I will put it probably. So it's encouraging. How long will the natural or vaccinated acquired immunity last? Right, so this is acquired immunity. So in other words, there was a time when none of these monkeys were immune. They were not immune. Then they had the disease. Study 1, they had the disease. Then they acquired the immunity because they'd had the disease. The other monkeys acquired the immunity by being vaccinated. So which will stay immune for longest? Will people who've actually had the disease remain immune for longer? Or will people who've been vaccinated actually remain immune for longer? And how long will these 2 groups remain immune? No idea. No one knows. No one knows that. So how long will the natural immunity from people who've had the virus, the actual virus, last? Or how will the vaccinated immunity last? They're both acquired immunities, but how long will they last? We don't know. Although, what we know about other coronaviruses, I'd be surprised if it didn't last for a year. And it could well last for a year to 2 years and probably longer. But I don't know that. I have no data to back that up. No one in the world has data to back that up. But what I suspect and what I hope is that the immunity from the vaccine would last long enough that this disease could be eradicated. That is the hope. Just as smallpox was eradicated in 1975, just as SARS-1 was eradicated in 2003. So that's a review of vaccines. Now, that's the end of this video. I've just got a couple of things to tell you, though. One is, we're going to look at some pictures of viewers in a minute, which is great. You're the important people. But what I want to give people the opportunity to do is record a short video and send it to me. I think we'll do it on a Facebook link. I'll put you a Facebook link of your experiences with this virus. Now, that could be that you had the disease, and then you might want to tell us how you felt when you were ill. That'd be great, because other people could partly know what they might expect. And we did this before in a video interview. Or you might want to tell us what it was like looking after a member of your family who was ill. Or you might want to tell us the difficulty you've had in terms of confinement or other problems that this virus has arisen. Because Stalin reportedly said 1 death is a tragedy, 1,000,000 deaths is a statistic, means nothing. So what we want to do is sort of, what I want to try and do is give you the opportunity to make this story human about your individual experiences, because that's what healthcare is about, really. It's about one person at a time. Now, that's not for everyone. I know that. If you send me a video in, a lot of people could end up watching it. So, if you're up for that, fine. I know it's not for everyone. If you don't want to, obviously, this is just if you feel. I would ask you to consider this if you feel your experience on a video would benefit someone else. So I don't care if it's interesting. I don't care if you're interesting. I'm sure you will be. But the only purpose in doing these videos is to help people. If you think it would benefit other people, then send me in a short video about your experience. Right, let's look at some of the important people. And we have, what have we got? Second screen. There we go. If we have the technology. We do. Right. This is Amy. And I think Amy might be in Colorado. So thank you for sending that in, Amy. Glad to see you're wearing a mask to protect other people. It's just a matter of time till people start advertising on face masks, isn't it? So Amy's advertising caution. So pretty soon we're going to have adverts for, I don't know, chocolate biscuits or supermarkets on face masks. Anyway, you and me won't make any money out of it. So thank you, Amy, watching in Colorado. This is Francis with Fergus the dog, watching in Scotland. And I think it might be Francis that's taking the vitamin D. Good. Pleased to see that. This is Jen from Vancouver, who's got a mask on ready for going out. Thank you for that, Jen. This is Jeanette and Simon in Lapland, Finnish Lapland. Now, it could be that these are the furthest north viewers to this channel. Could well be. So thank you for watching in Lapland, Jeanette and Simon. This is some artwork from Joe in California. Wear a mask so others won't need to wear a ventilator. Powerful point, Joe, powerful point. Thank you for the artwork. Now, I think this might be Joe as well. So glad to see the vitamin D, the soap to clean his hands, the pulse oximeter just in case, the masks, and even the mannequin is wearing a mask. And you're watching my video, so excellent all round. This is Juiced in the Netherlands. I think Juiced is doing some sort of master's degree in science, so that's encouraging, and glad to see Juiced is also watching the videos in the Netherlands. Do hope you come up with something, Juice. We need scientists. This is Rais watching in Cyprus, down there in the island of Cyprus. This is Ryan watching in Hong Kong. Great to know people are watching in so many parts of the world. So we've ranged from Hong Kong to Lapland today. Brilliant. This is Stuart from the US of A. Looks like Stuart's a musician. So thank you for watching in the States, Stuart. This is Tasmin, and I don't know what your name is. So I know it's Tasmin and someone else. So thank you for sending it in anyway, and thank you for watching the videos, both of you. In Sussex. To me, that's southern England. Oh, this is Tom, who walks and watches in Ohio. So good to see Tom's getting some exercise as he watches.